Okay, this is the second portion of our protein lecture. So at the end of part one of proteins, we learned about the various functions that a protein can have. And now this statement right here is going to be really key for you to understand not only the rest of this chapter coming up, but also for many, many different concepts that we're going to talk about for the rest of the semester. So the function of a protein is determined by its shape. In fact, I feel that this statement right here is so important that I'm going to put this question right here on the lecture test. I want you to know that the function of a protein is determined by its shape. Say it out loud with me now. The function of a protein is determined by its shape. So, all of the different functions of proteins that we just talked about are all going to be related to the protein shape. And in order to get to the shape of the protein, we're going to be looking at 4 levels of protein structure. Now, the first level of protein structure is primary structure. And this is literally just the sequence of amino acids. Recall that we put these individual amino acids together using dehydration synthesis, forming these peptide bonds. And so think of the primary structure as just a long linear polypeptide of these individual amino acids. Now, secondary structure is seen because of the hydrogen bonding that occurs along this polypeptide backbone, and it causes the polypeptide chain to twist or fold. So recall what hydrogen bonds are. These are these bonds, weak attractions between a partially positive hydrogen ion and something that's partially negative, like this oxygen atom here. Individually, the hydrogen bonds are very weak, but collectively they're very strong. Now, secondary structure can exist where our polypeptide chain is twisted and coiled, forming something called an alpha helix, or where the hydrogen bonds form and make a beta pleated sheet as the polypeptide chain folds back and forth on itself, and you can see these hydrogen bonds here. So hydrogen bonding is really, really important to secondary structure in our proteins. Now, tertiary structure is where we see interactions among these R groups. And so the R groups, remember those are the 20 different variables that make up the 20 different amino acids, causes additional folding and produces a truly three-dimensional molecule. I want you to visualize these proteins as literally three-dimensional molecules. And so if we look here, this purple line represents our alpha helix, and you can see here's an R group and here's an R group attracted to each other because of their hydrophobic properties. So you can see how this creates a fold. Here is another R group and another R group attracted to each other by a hydrogen bond, again helping to twist and fold this secondary structure polypeptide. So there's lots of different things that can cause the twisting and the folding of tertiary structure to help give this protein a really three-dimensional shape. Now, quaternary structure occurs between 2 or more different polypeptide chains. Many polypeptides or many proteins like hemoglobin depicted here are made up of more than one polypeptide chain. So we see a polypeptide chain going from primary to secondary to tertiary structure, and then it joins other tertiary structured polypeptide chains to form our fully functional protein. So for instance, this is our hemoglobin protein. You can see here the heme or the iron atoms that will allow this particular protein to carry oxygen. It looks just like a little donut or a nest, and that oxygen atom will fit right inside of here. Now again, the function of proteins are determined by their shape, and their shape is crucial to their function. So when a protein changes its shape, it also changes its function. Think about the gelatin protein that we used in lab. When it starts out, it's a powder. You mix it with some water and heat it up, and it becomes a clear liquid. As it cools, it crystallizes. Each time that protein's shape has been changed, and ultimately its structure will be changed. Now, what's interesting about many of these proteins is that we use a word called denatured. that talk about how a protein's shape may change, and it may cause the protein to actually lose its functionality. So, for instance, some proteins are shaped, have a three-dimensional shape, such as this, and then when they get heated up, they lose this three-dimensional shape. Now remember that heat can strain those hydrogen bonds and it causes the protein to become denatured. It's this denaturing that is one of the reasons why if you're going to cook a roast, you cover it with water and you cook it on a very, very slow but low temperature for a long period of time. That heat and that water helps to denature some of those proteins that hold those muscle fibers together that make that roast so tough. If you were to just cook it quickly, it would be really hard to chew, but by the time you've left it in the crock pot for 5 or 6 hours with that heat, all these proteins have become denatured. So as we move forward, we'll see how denaturing or changing a protein's shape can inactivate or sometimes activate a protein. Some things besides heat that can help denature a protein are pH, acidity or alkalinity, temperature, pressure, etc. can all help to change the shape of the proteins. But recall that these proteins are really large three-dimensional molecules and their shape is crucial to their function. And when their shape changes, their functions change. So be sure you review the 4 levels of protein structure. This slide right here just kind of summarizes protein's primary structure with the sequence of amino acids forming these peptide bonds. And then we have secondary structure, either the beta pleated sheet or the alpha helix, leading to tertiary structure, this huge three-dimensional molecule. And then we join these polypeptides together in quaternary structure to make our fully functional protein.